It's my pleasure to introduce our next speaker for today, Dr. Sandy Raha, who is one of our own. He's an associate professor here at McMaster in the Department of Pediatrics. He completed his PhD in biochemistry at the University of Toronto, postdoctoral studies at the Hospital for Sick Children in the area of metabolic diagnostics. We then lured him to Hamilton, where his research has focused in large part on the impact of maternal use of cannabis during pregnancy, the effects on the mammary glands and and the placental function, and how that affects the health of the baby postnatally. And he's here today to talk to us about the effects of cannabinoids on the placenta. So thank you, Doctor.
Thanks very much, Jason. I'll just wait for my talk to... So I changed the title up a little bit from what's in the brochure because I really wanted to focus on two areas of study in our lab, the mammary gland and the placenta. Now, to quote an old Monty Python cliche, and now for something completely different, I'm going to be focusing on a very mechanistic approach, which is what we take in our lab, looking at trophoblast cells in the placenta and most mammary epithelial cells in vitro. So, oops, am I hitting the wrong button? There we go. Is it possible to get that? Okay. Apparently I fail amongst the first three presenters. As we heard, and I kind of was a bit lazy and took advantage of the fact that I knew several presenters going before me would be providing a very thorough review of cannabis and cannabinoids. I jumped right into the information that, we all know that the endocannabinoid system is very widely distributed within physiology. It acts through two primary signaling molecules such as anandamide and 2-arachidonylglycerol, 2-AG. Now, it's very important to keep in mind that these are the natural targets of the endocannabinoid system, and THC and CBD are really exogenous sources that compete with these endogenous ligands. The system also consists of a series of synthetic enzymes and degradative enzymes, as well as receptors, and Dr. Hardy summarized that very well in his talk as well. So we can go to the next slide. Thank you. So we know that cannabis has a consequence of use during pregnancy. You're going to hear a lot about the epidemiology and the clinical manifestations, but I just wanted to prime everybody with the fact that, and you've heard this before, that the use of cannabis during pregnancy, it's fairly evident that it's linked with decreased fetal growth, low birth weight. There is an increased risk of premature birth that's linked to cannabis, and there are postnatal neurodevelopmental issues that we see in babies exposed to cannabis in utero. So moving to the next slide. Oh, I see what happened. It's because I used a PDF and not this. Okay, sorry about being the deviant there. So I just want to. So the approach in our lab is a little bit different. We decided we wanted to understand the mechanism, so we started with some in vitro experiments. We are working towards an in vivo model, and I'll finish off with a little bit of a description of that. But we're really focused on two primary areas: the maternal-fetal interface, the placenta, and also knowing that Cannabinoids are very hydrophobic. They get sequestered to fat tissues. They end up in the breast and subsequently in the breast milk. And cannabinoids can exist at almost 8 to 10 times their serum concentrations within adipose tissue. So those concentrations can then leak into the breast milk. So for that reason, we focused our mechanistic understanding on those two tissues. In the placenta, we know that there are effects on the trophoblasts. These are the primary stem cells in the placenta. They go to make up the maternal-fetal interface, which in the human is actually one cell layer thick. So we want to look at the consequence of forming that, consequence of THC and CBD in forming that single layer, single cell layer, the maternal-fetal interface. And we want to look at the consequence of THC and CBD in formation of milk proteins in mammary glands. And understanding those mechanisms, we're going to bring that together in our animal model, which we're working on right now. So if we can go to the next next slide. Thank you. Oh, okay, great, thank you. So as I was saying, the maternal-fetal interface in humans is one cell layer thick, and you can see in blue there the formation of this maternal-fetal interface is really determined by the fusion of these precursor, what we call cytotrophoblasts, into a single syncytium. So this is a multinucleated cell layer, and that forms the actual interface and is responsible for transport of gases, transport of nutrients, release of hormones. That's its function, to sustain and help the fetus grow. Perfect. So in this paper that was published by Dr. Hardy a few years back in 2019, he found a very interesting phenomenon in these cytotrophoblasts. He found that treatment with THC caused a decrease in mitochondrial function, caused an increase in endoplasmic reticulum stress. The endoplasmic reticulum is responsible for the formation of proteins and processing of proteins for use in the cell throughout its life course. So seeing this and recognizing that there's some very old literature pointing to mitochondrial dysfunction and THC in the literature, but that hasn't really been followed up recently, because the primary focus of cannabis and cannabinoid research has been on neurodevelopmental outcomes and looking at classical cannabinoid signaling pathways through the cannabinoid receptors. And so very recently, groups like Dr. Natales and Dr. Hardy and our group, we've been trying to focus a little bit more on the metabolic outcomes and some of the more classical intracellular signaling processes that are lesser associated with cannabis. So seeing this paper, we were already working on some of these pathways within our lab, but it sort of gave us cause to celebrate, because we knew things were going okay, because we saw these profound effects of cannabis on these cytotrophoblasts and syncytiotrophoblasts on their mitochondria. So mitochondria, very important for cellular bioenergetics, and the endoplasmic reticulum. So this is some work done by a former PhD student, Valencia Walker, who showed that treating these cells with THC resulted in poor syncytial function, so reduced syncytialization. And that's shown here by the green staining. If you look at the first panel, the one on the top is just our vehicle, methanol. And that shows normal fusion, so that you see very you see reduced amount of green, which means the cells are fusing. The green color outlines the border of the cytotrophoblasts by staining a marker called E-cadherin. And you see much more green in the samples treated with THC. And so within these cells, we also noticed there were increased markers of cellular stress proteins, some classic chaperone proteins, which are responsible for helping cellular processes recover when proteins are misprocessed, called HSPs. These proteins were increased in the cytotrophoblasts, and we also saw reduction on the panel on the extreme right bottom. The total ATP production within these cells were reduced, and we also saw increased formation of free radicals. We saw increased formation of enzymes that are responsible for mitigating the stress caused by free radicals, called superoxide dismutases. And interestingly, despite this compensatory response to the free radicals, we still see cellular damage. Haven't shown that figure, but we do have evidence of cellular damage. That means that THC is causing this increased level of oxidative stress, and that might be one of the mechanistic routes to cellular dysfunction. So we tried to link this to a clinical outcome by looking at some of the growth hormones synthesized by the syncytial cell, the syncytialized cells. And what we found was that when the cells are treated with THC, we see reduced amounts of placental lactation and IGF-2. Both of these markers are linked to fetal growth. Interestingly, we also find CBD. Treatment with CBD results in a decrease in ATP production, so that OCR, the oxygen consumption rate, measures respirometry within these cells. But the interesting thing here is we haven't been able to see the degree of increase in free radical production or superoxide dismutase response that we saw with THC. However, we do see reduction in cellular differentiation markers. We see some of the compensatory increases in the HSPs, but it's not consistent. We don't see them as many of the HSPs as we do for THC, and we seem to see reduction in the markers, the gene expression markers for cell fusion. So our hypothesis is that both THC and CBD are decreasing the cycle of single cells fusing to form this syncytium. And that is one of the areas where the two compounds are impacting. And part of that role, so part of the mechanistic contributions there are through ER stress and mitochondrial stress. So just to the second part of the talk, where we had the opportunity to work with Dr. Alison Shea to look at some of the impacts of cannabis use during pregnancy on breast milk. Now, we were interested in, so I show this diagram to show that the mammary gland undergoes a phenomenal amount of developmental remodeling during pregnancy. The biggest changes sort of happening at puberty and then at pregnancy. And so we focused our studies on a murine cell line, which was, it's called a human mammary epithelial cell. It's the cells that line the lumen of the murine mammary gland. However, we first went to the human data and we did a very small clinical, a pilot study with roughly 19 women who 19 cannabis users. And one of the things, one of the first questions that we asked was, could we detect cannabinoids in the breast milk of these users? We had 13 women who used cannabis who anecdotally reported to us they used cannabis during pregnancy. 8 were pregnancy and lactation. Sorry, 1 during pregnancy only, 8 during pregnancy and lactation, 2 lactation only, and 2 women we had no information on. But when we looked at the levels of cannabinoids in the breast milk, we found a median concentration of about 22 nanograms per mil. You can see what the range is there. We also found levels of the metabolites of cannabis hanging around in the breast milk. So what's going on? How are the cannabinoids affecting the mammary glands? One of the outcomes that we did find that was significantly different was the serum IgA levels. Now, IgA is an immunoglobulin that's responsible for imparting early immunity to the fetus, to the baby, to the neonate. And that seemed to be reduced in the cannabis users. We had a small number of individuals who were that use both cannabis and cigarettes. This is often a complicating factor in any type of clinical trial with cannabis. We tried to separate those out. And we also analyzed macronutrient composition. In this small sample size, we really didn't see a difference in the macronutrient compositions. So we went to our cell culture model and found that both THC and CBD treatment of these murine epithelial cells resulted in reduced gene expression and reduced protein expression of 2 of the primary milk proteins, beta-casein and whey acidic protein. You can see that THC doesn't significantly reduce either one of them, but CBD does. And so this was quite interesting to us. Furthermore, we wondered if we see the same kinds of impacts on mitochondrial function and ER stress that we did in the placenta. And when we examined markers, gene expression markers for ER stress, we saw that both THC and CBD significantly upregulated ER stress markers. And what was interesting was treatment of these cells with 4-PBA, which is known to attenuate ER stress, resulted in normalization of the production of milk proteins. And so that's been quite exciting, and we're obviously following up with further studies on that. And with that, I will conclude and thank the people that did the work. Olencia did a lot of those original experiments with THC. Chipman did the clinical, help with some of the clinical work and did the work on the mammary gland. And Tina is currently working on some of the placental outcomes. And we are in the process of building a new a murine model of smoke exposure with cannabis. So exposing mice to cannabis smoke and looking at placental and mammary gland outcomes in those tissues. So stay tuned for more exciting results. Thank you. I'm happy to take any questions.
Hi, Sandy. Really exciting presentation. Just to relate the stuff you saw in the placenta with the breast tissue or the cells, do you think THC or CBD is invoking ER stress in the breast tissue?
Yes. So the THC. So we did gene expression markers in the cells. We are looking. I think we will find those markers also upregulated in the tissues. We have both placental and breast tissue from our model, and we're going to follow up to quantify those markers in the tissues. But I think there is ER stress going on. Whether it's the primary mechanism or not is the million-dollar question. But we know that especially mitochondria are linked to, are great thermostats of cellular stress. And they serve to either preserve the cell or tell the cell to die, apoptosis, based on a level of stress. And so I think that the exact contribution remains to be elucidated, but I do think, and mitochondria and ER stress are both intimately linked. So I think we will see both in tissues. And this might be a general phenomenon because some of the neurodevelopmental outcomes, I think, will also show mitochondrial and ER stress-related mechanistic contributions. Thanks, Dan.